have got through a lot of stuff now. We've done most of the basics, and we had a go at RNA-seq. The next technology we're going to look at for high throughput sequencing and how we can analyze it is ATACseq. So anyone here going to be doing ATACseq? You guys are going to be doing everything just as well. So ATACseq is a great method, and it's kind of developed more recently than most of the high throughput sequencing methods we use. And actually, it's one of the ones I had the joy of discovering how to analyze when I came to Rockefeller. So I had to kind of pick this up and learn this when I came. And what we did is I actually hosted a little class on there we go. Actually, the top hit. So we did a little class on New York R and Bioconductor. I'm trying to find the actual link to it now. There we go. Oh, so complex just to find it. Anyway, so I took a lot of the public data. So I took the original course, the original ATACseq data, picked a little bit on Christina Leslie again, and we took some of her data again, just so that we can get a good idea what the original ATACseq looked like, what the modern day ATACseq looks like, and really dig down into low level looking at all these metrics. So that's what we're going to cover today. I've split it up into 4 or 3 sections. A lot of this we've already covered, you know, basics, but now we're applying it, so hopefully we can put it into some context. So ATACseq, I think it stands for Assay for Transposase Accessible Chromatin Using Sequencing, uses a transposase to efficiently fragment accessible DNA prior to sequencing. So this should provide a map of accessible open chromatin genome-wide. So I'm not sure if anyone, I'm not sure how much of your epigenetics you know yet, but obviously accessible chromatin is chromatin which is not compacted. And actually ATACseq is really going for very open chromatin. And from this method, by finding regions of open chromatin, maybe differential regions of open chromatin, we can then make assumptions about what sits in that open chromatin. And that can be in combinations with ChIP-seq or by using motif hunting. And actually, there are a lot of tools about finding open regions of chromatin. If it's differential and it's only in one condition, maybe it's interesting. And then we can look at what the motifs are underneath. In contrast to other techniques which promise to do something similar, ATACseq had a lot of advantages. And one of the major advantages, I'm not in the lab, so it doesn't affect me, is that you could do it in a day, which is very, very appealing, especially if you're doing large numbers. Maybe you can do 2 or 3 batches in a day. And you need a very low input material. So compared to things like ATACseq, which is now probably a dead technology, but DNA-seq too, you need a lot more material for that. So, you know, I think a lot of you would know this, but given closed chromatin and open chromatin, euchromatin, we can use this TN5 transposase, and what it does is it breaks up the DNA in the areas of open chromatin where it can go. And this really means we're going to get lots of different types of fragments from our sample. But really, we're going to be interested in 2 types of fragments. Fragments of totally open DNA, so regions like this, and regions around nucleosomes. So if you look here, this nucleosome isn't compacted into closed chromatin. It is open, it's an open chromatin, and we will get with this TF5 transposase cuts either side of this nucleosome, this histone component the DNA is wrapped around. And then eventually we're going to end up sequencing that. Right? So there were, as I said, this is a great great image. I've taken it from someone else, so I need to probably create my own version of this image. But just to give us an idea of the different types of techniques doing something similar, DNA-seq was the enzymatic digestion of open chromatin, really around transcription factor binding sites. And if we have our little model of DNA here, we have our nucleosomes, our histone complexes, and then we have probably, let's say, an enhancer around a transcriptional start site. We have some open chromatin, and we have a transcription factor sitting in the middle of this. When we did DNA-seq, we would actually extract sequence from around this open region and around this transcription factor. And we could use the size of the fragment, right? This fragment's bigger than these smaller fragments to give us an idea of where the transcription factor is. Aminase-seq did something slightly opposite. It was really looking for breaking up the nucleosomes. You didn't get signal from this open region in the middle, but you got signal around nucleosomes across your whole genome. That's expensive, effectively mapping your whole genome with Aminase-seq. So something my wife tried to do, and it just costs a lot of money. ATACseq actually allows you a method to simultaneously extract signal from both this open region between nucleosomes, and then to a lesser extent, you're going to get reads from around nucleosomes. And the more compact the chromatin is, the less reads you get. So we should be able to, with this, get the position of nucleosomes, maybe, right, from this type of signal, but also get a map of where the open chromatin is, where our transcription factors may be binding. We might even get some signal kind of reflecting the area which was protected because a transcription factor was bound to the DNA. So this is all cutting around areas which aren't covered or protected by other features on the DNA. So we're going to cover the basics today, ATACseq basics, and we're going to look a little bit post-alignment too at some quality control we can get from some simple tools. I actually think we're going to plot this at the beginning of our next session. So it's not. So like I said, when I first put this course together, I spent a lot of time making sure we had some great data. So I think I took it from 3 data sets. This is the original ATACseq paper, I believe, by Buenstro and Greenleaf. He's gone off and made his own lab. Buenstro does new stuff, and Greenleaf has maintained their tools in ATACseq. We can download this, and we could go probably here, which will be the ENA browser, and we can go download this one sample, or the samples associated to it. I've already done this, so we don't need to. Oh, that's the wrong place. We don't need to. The second data set is going to take from ENCODE. They have lots of data, and most importantly, they have 2 replicates for every group, so we like to use them too. And the third data set, again, is from Christina Leslie's lab. And if you remember, in the RNA-seq, we were doing some of the same kind of experiment. So we were looking at T regulatory cells, naive, before they've been exposed to some stimulant, and then activated. They've been presented with the antigen. We can find FASTQ for those files here and here. We're actually going to be working with the aligned data. So I've already done what I'm going to present in the course, some of this data. But it's actually on ENCODE as well, I believe. It's just a much newer data set compared to some of the other data sets with it. So Bing Ren, maybe that's a while ago. Yeah, maybe. Same thing. So we need 3 pieces. We need some reference data. We're going to need a reference genome in FASTA format. We know where to get them from. We can use the BS Genome Bioconductor packages or your favorite FASTA file of genome sequence. Gene models, we can get those from the TXDB packages. We've seen that before. We can also get them from a GTF and turn it into a TXDB. And finally, we have something which we'll talk more about in our ChIP-seq. This is the blacklists. I'm not sure if anyone knows about the blacklists. It's fascinating. See if we can, what it pulls out if I go to the ENCODE study. See, maybe I didn't follow quite the right link. Try it again. Oh, didn't go. Do you have any history on it or anything, or a PubMed link maybe? Okay. So I did a lot of work with these actually in my first years as a bioinformatician because I was very big on ChIP-seq, my speciality. We wrote a little paper on this too. And really it was a fantastic piece of work by a number of labs, but Anshul Kandarji, I think we can probably say, was the guy who really put it all together. And what they did is fascinating, and we can actually see it better in the ChIP-seq. But what they did is trawl through all the ChIP-seq, which was coming out of the ENCODE study, and they started to see that there were regions of the genome which just had signal no matter what you were trying to chip for. So if you're looking at MYC, CTF, polymerase chip, there was always signal there. And actually, it turns out from our work and other people's work, it turns out this is basically to do with repeat expansion. It's because your genome's got bigger than you think it is. And if a genome which you think is 100 base pairs long is actually 100,000 base pairs long, when you go to get a random sampling of reads over that region, randomly sampling over 100, you're not going to get many reads. 100,000, by chance, you get a lot of reads. And then when we realigned it back to the genome we understand, we end up with this tiny bit of region, 100 base pairs, which is probably repeated 1,000 times in the genome. And we map to that one region and we just get a pile up of reads. So he went through and he actually paid a lot of attention and manually effectively selected these regions, and now they're filtered out from most ChIP-seq. We're going to cover it more in the ChIP-seq course, because the reality is, I used to work in Cancer Research UK in the UK, obvious. But if you work in cancer cell lines, those cell lines change all the time. The blacklist changes all the time. And you need tools to effectively discover your own blacklist. So there's something called gray list, and we'll cover that in future sessions. But using the ENCODE's derived blacklist, we're going to do a good job about removing much of this artifact. So the blacklist is really just per genome, a set of regions where we believe we have artifact signal, and we're just going to filter them. So to get started today, we need a bound file and binary files, obviously. We're going to show you how you align them, but I've already produced them, and you can download them from here and here directly. If you want to get, and this has a little bit more because I did this originally as a separate course, but if you want to get all the files you could possibly need to follow along, you can click here. If you want to get all the intermediate files and everything, you can click here. And that's going to get all the bound files and everything you would possibly produce in this class. Bigwigs, if you just want to follow along in IGV, you can download from here too. Now we can get started. So prior to alignment, we always recommend checking your FastQ quality. Some basic QC checks may identify bias in your data, so you have GC bias. So we'd already seen this kind of in our Bioconductor intros. We saw it in our RNA-seq, and we're going to do it again in our ChIP-seq. So I thought I could save us some time, and we'll hold off and go look at the FastQ quality checks in our ChIP-seq. Reality, you can run FastQC, just see those plots, review those plots we talked about, and you'll be okay if nothing looks too strange. So we're going to work with Greenleaf actually through this course because it's a really great example. We have his plots from his paper, so we can compare everything we do, and we can make sure that we can recreate the original ATACseq. First thing we need to do is create something we can align to. We've done this before, we can do it again now. We're going to create a FASTA file of the major chromosomes, and we're going to write to that, and that will be our input for our index. So we'll just do library, BS genome, H sapiens, UCSCMM10, HG19. So that will load this object into memory. Then going to quickly make a vector of all the chromosomes we're interested in. I loop through that vector, and for every time I go through the loop, I extract one of the chromosome sequences, and that comes back into a list. with that list, I will then provide names, because the names I provide, which will be chromosome 1 to 21, these are what goes in the FASTA header line. So I need to provide names. Once I've got my named list and each element is a DNA string, I can then turn that into a DNA string set. And I can write that out to file here with writex string set, and that will provide a FASTA file, which is all the sequences for the human genome. If, for whatever reason, you don't want to use the FASTA files from inside the BS genome object, you can just start from whatever the FASTA file you have here. And that includes, downloading it from Ensemble or iGenome. We then use our build index function from the subread package. So we're building, this is just our FASTA file. We then need to build an index to align to. And to build an index, we're going to use the R subread function build index. We'll give it our FASTA file, which we just created. So from here, we're going to tell it the name of the index to create. And then that's going to create a file called whatever this, main cur. And then we'll set here now a few features to control memory. So first off, we'll tell it index split. And that means it's going to try and chunk up our index into separate chromosomes or separate regions. And then we'll set memory, and this is going to be the amount of megabytes used both in the indexing, but also the alignment. Right, so you set this low. When you come to alignment, you're also going to save memory. If you set this high, it's going to get faster, but you're going to need more memory. So I think by default, it sets this at 8,000, but you always need to set index split to be true or false. So here, if we set it to true, it's actually going to try and reduce our memory. If you set that to false, this memory parameter has no effect. So now we have an index. We can align our ATACseq reads. But ATACseq data is a little different to what we've been looking at previously. Right? So RNA-seq data can be single or paired end. So can ATACseq data, but more often than not, it is paired end. And there's a real reason, as you'll see as we go through, why paired end sequencing became kind of popular for ATACseq. So for paired end sequencing, the most likely, I mean, I think 99.99% of the time, going to see your reads kind of separated like this. So you'll have a sample name. So this is the name from SRA. But then you'll have either an _1, _2, or an _R1, _R2. And that just tells you that these reads are paired. This is the first read in the pair. This is the second read in the pair. Okay. So paired, well, these two matched paired end read files should contain the same number of reads. They may not do. If you've got them straight from your genome center, they will do. Someone may have filtered or changed your FASTQ before they get to you. Okay, so that is a possibility. So you might want to check that. But we can get an idea of what's happening inside our FASTQ pretty quickly just by loading the short read library. So short read allows us to handle FASTQ data. I can then read the first pair here, the second pair here, right? And I know it's the first pair by _R1, _R2. And now if I actually look at the ID for my reads using the short read ID function, I just look at the first two read names. And it tells me a little bit about the width, whatever, but I can see here the machine, flow cell, the position. And then here I've got this 1. If I look here, I've got the equivalent 2. And then this name here should match everything else in this file. Oh, sorry, in the first ID. So this ID and this ID match. That means they were paired. Can you see that? So this name here and this name here, they do match, right? Yeah. Yeah, the only difference really is this, read 1, read 2. So these reads come from the same fragment, and they're actually reading from either end of the fragment. So we can look at that here. If we look at, this is the actual fragment we're reading, okay? There's a little bit of a confusion when you go to your genome center. You're going to have difference between fragment sizes. Remember, when you go to your genome center and you get that final size of your fragment, that often includes the Illumina adapters to the end of your fragments, and they are just required for sequencing. That's where we start sequencing from. But it's this insert, this fragment, which is our arbiter sequence. So this is these broken up bits of DNA here. Those are what we're sequencing. And then we'll add the adapters, and then read one will start from one side and read into the read. And then in the sequencing, we'll flip this, and then we'll read from the other side, going backwards across the fragment with read 2. So you can imagine quite often read 1 and read 2 may overlap in the middle. And they're short enough the fragment. We'll get an idea of this. We should have sequence matches. But this allows us, this gives us a lot of power in sequencing, because I can align this one, and then I can align this one separately. But then by knowing that they're from the same pair, there's an expected distance between them, right? Something between 0 and probably 3,000 or something. The distance between these two reads will also allow me to map better. So then we have what we would call the fragment size is often considered with the adapters. That's often what you'll see when they run a bioanalyzer or an electrophoresis gel. You'll get the size, and it will include the adapter. And then the actual thing we're sequencing is often the insert, and we'll refer to the size of that as the insert size. Cool. Just the read 1. So it's this. So that bit in black here, that's what we're reporting for our first read. And then the second read will report this going this way, right? But backwards. Oh, because these are the read names, right? So this read name is the same across the two, but the sequence will be different. It may overlap in the middle, and then you'll be able to tell this. But yeah, this bit here is the ID. If we did seek read 1, we would get the sequence for it. And actually, we wrote some software which, when they overlap, it allows you to make one long super read, and that's useful for it. We can use that standard alignment of DNA as we did for ChIP-seq, but we need to increase the maximum allowed fragment length. So what I was saying here about the maximum distance between read 1 and read 2 can often be a little bit bigger than we might expect for ATACseq. So that's one parameter we want to change in our normal alignment versus what we saw in the standard bioconductor. So I'll do align, and I'm going to set the BS genome index we want to align against. I'll tell it, I've got 2 reads now, so I'll tell it read 1 file is read 1, read 2 file is read 2. And that's just the names of the files here. Output file, we're going to call it ATAC50. This is the name of the sample on GEO. I'm going to tell it type equals 1. Type 1 means DNA here. We didn't have to change that because we used subjunct for alignment previously. We're going to tell it number of threads. So strangely, subread isn't quite bioconductor in its parallelization. So we can't use BioC parallel to set it up, but we can say number of thread equals 2. We'll say unique equals true, so it's only going to report uniquely mapping reads. And then we're going to say max length equals 2000. Okay. There is a min fragment length as well, but we don't need it here. But we're saying consider a read properly paired if it lands within that distance of its pair, read 1 and read 2. We can also use our bow tie to do the same thing. So just to show you that, and a lot of people like bow tie 2. To do that was very similar. We just do library rbowtie2. We can provide the FASTA file we produced to references. And then we just tell it the name of the index we want to make. So here, the same as previously with underscore bowtie2. And then if I want to do an alignment, the pain with Bowtie 2 in R is that I need to unzip the FASTQ files. It doesn't take the zipped files, so I use gumzip function, which will just unzip a file to just the FASTQ. And then if I wanted to use this aligner, I will then use the Bowtie 2 function, provide the index, provide the SAM output. So it's going to make a SAM file rather than a BAM file. And then I will provide the links to the two FASTQ I just gzipped in this step. We don't need to specify a maximum fragment length in Bowtie 2. It's not going to cut off reads if they're too far away. And then we can just do, on our SAM file, we can just do as BAM, give it the path to our SAM file, and it will just update that file and produce a dot. And so either way, we can either use standard R subread, which I prefer, I understand a bit better, or we could use Bowtie 2, which is also a very popular tool for ATACseq. I think they use Bowtie 2 in the paper, which is why I use Bowtie 2 here. Not sure if we've used this before. Have we used unlink in the class before? Probably deliberately not. Unlink means delete, so be careful with it. So what we'll usually do is, this is your script. I'm just putting it here to make sure that when you've done your alignment and you've made your BAM file, remember to delete the SAM files and delete these FASTQ files, because they are going to be huge. SAM file is wasted space. You have the same information once you've done as BAM. It's created a BAM file. So just clean up your scripts for Bowtie 2. Following alignment, we want to probably do our ordering and sorting. So here we're going to use our SAM tools again. We'll just create the path we want to our sorted BAM file. And then I'm going to do sort BAM. And I'm going to use my out file from probably here. And I'm going to pass that now to sorted BAM. And then we'll give it what we want to create. I'm just doing this G sub because I'm removing the .bam extension. If you remember, when you run sorted BAM, sort BAM, it doesn't want to have the path you to the file you're outputting should be the prefix. It should never end in .bam. Otherwise, it's going to create a file called sorted bam.bam. We can then index our file, and indexing is super important because it makes our file read access, random read access, so you can read from anywhere in your file. It means it will work in IGV, and it also means we can get those very quick stats from IDXstats. We don't need to read the BAM file, we can just read the index to know how many reads we have. So there are some really simple QC we can start to do, and I think this first one is the one in the paper and is the first thing you ever need to check in ATACseq, and it will tell you whether your experiment has failed by a very common problem with ATACseq. So we need to check the distribution of mapped reads across your chromosomes. We know IDXstats doesn't provide much information, but it does tell us how many reads map to each chromosome. So we can use that now because we ran index BAM here on our sorted file. So we can just load the RSIM tools library. It should have been loaded here anyway. And we'll do IDXstats BAM on our sorted BAM file. And that will produce for us a data frame of all the chromosomes and the number of reads mapping to them. Okay, so show you what that looks like. Guess I have a BAM file somewhere here. Session, set working directory. Session, set working directory, choose directory. Oh, actually, maybe I didn't download the Sure, I've got one in here somewhere. Library of samples. And then, stats, BAM, data. Let's do heart. Oh. I don't know what I just did, but I just blew up my arm. So maybe we'll. I get the feeling that that wasn't a complete BAM file. So maybe I'll try just the demo for now. If you ever get that, like your arse suddenly breaks like that, you've either hit your memory issue or it's because something you're using doesn't necessarily error well. And it's usually C++. So it's passed it down to C++ and some memory's going around. No big consequence, just to restart the file again, I guess. Sorry, I was looking for an example of IDX stats BAM, but I don't think we have one in there. Things here, so. Index BAM. Oh. That was painful, but so we have seq names, so it tells us the names of the chromosomes, tells us the length, and importantly, it tells us how many reads were mapped on every chromosome. Okay, so that's really what we're after. And once we have that information as a data frame here, we can then pass that to Ggplot and just get a very quick overview bar plot of where all our reads have gone. So if I do library Ggplot, I'll do Ggplot map reads, I'll do rows here will be the seq names, the chromosomes, the number of mapped reads, I'll fill them in by the chromosome name. We'll do G on bar, but I'm not asking it to calculate the size of the bar, so it's stat equals identity, and then I'll just do toward flip, which just rotates the flip plot. So plus toward flip just literally rotates your plot 90 degrees. And already we can see something really weird. Right? If we look here, chromosomes are in order of how big they are. So it makes sense that chromosome 1 was the biggest up until around here, chromosome X. Chromosome X is actually quite big. But most of the reads have gone to the mitochondrial chromosome. Right? This was commented on in that original paper. We have a lot of mitochondrial reads, but it was most of the reads. Their lab has managed to clean this up. And there's things like ATACseq, which reduces this greatly, this amount of mitochondrial reads. I can tell you it's still high though. So you still are spending a lot of money on this mitochondrial read. And actually for the rest of ATACseq analysis, 99.9999% of analysis, we throw away the chromosome. It's a circular chromosome. It's very hard to analyze for mitochondria. Right? But that's a lot of reads wasted. So this is the first thing you can check to see if your ATACseq worked. It's the first thing people ask me to check for them. And then hopefully with the new methods on the attack, etc., you shouldn't see this much result. It's also a QC metric for single cell attack as well. So now we have processed the Greenleaf ATACseq PED data. We can start to work on the alignments. One thing we want to look at pretty quickly is the expected fragment length. Right? So given that we know we're going to be breaking up around nucleosomes and we're going to be doing an open chromatin, we can expect to see fragment lengths the size of open chromatin in there. And we can expect to see fragments the size of nucleosomes in there. So just to remind us, that's because we should have regions coming from around nucleosomes. We should also have regions coming from inside. Well, this is the nucleosome sized one here, from inside open regions. So these are also a very important QC for us, and they were important QC for that original paper. One thing we're going to do right from the start is going to set our flags to only read properly paired data. So we'll do library genomic alignments, because that's where all our tools to handle BAM files are. And we'll set a flag from our scan BAM param. So we'll get scan BAM flag, and I'm going to set one flag, and I'm going to say a read needs to be properly paired. And that means it needs to fit within this range we set earlier on. 2000 was what we set as the maximum fragment. So now I will build, now I have scanban flag, I can then build my scanban param object, and I'll provide the flags I just created. I will tell it, I want to just look at one chromosome here, so I'm just going to look at chromosome 20 from the beginning until this is the length of chromosome 20. So I'm going to provide that for the which. And then what do I actually want to pull out? I'm going to get the Q name, I'm going to get the I size, and I'm going to get the map Q. Okay, so Q name is going to be the read name. Map Q will be the quality of alignment. And I size will be the insert size of our fragment. So how long is our actual fragment? Okay, so that's going to be, come back, this size. How long is this fragment we actually sequenced? Right? And we can find that out from knowing where read 1 and read 2 are aligned in the genome. Okay, so now I can use my scanban param, and I've created a scanban param object. And then I will pass that to my readj alignments function, except now, previously we did readj alignments. This time I'm doing readj alignment pairs, giving it my sorted BAM file, and I'm telling it the parameters. I want this information. It's only over chromosome 22, and just give me properly paired reads. We get a GA alignment pairs class. We had GA alignments class before. And if we start to look at this, this is like an extension of our GA alignments we saw in our bioconductor classes, but also it's really like an extension of G ranges. Okay. And it's kind of cool. What it has is the chromosome it aligned to, the strand it was on, and then it has 2 ranges per row. And this is your read 1, where that mapped, and this is where your read 2 mapped. Okay, so it holds that all in one line for us. Okay. If we want to get into this and actually find out, if I want to look more carefully at read 1, I can extract my first read from my object of reads here. I can do first, and I will get the first read. Second, I will get the second read in the pair. So I'm basically extracting this read in the pair or this read in the pair. And then I can just look at read 2, and we can see we now have all this information I wanted. This looks much more like previous GA alignment object. I have a cigar, I have the Q width, the actual width, the start and the end, the junctions, but I also have the Q name, I have the map Q, so the quality score per read, and I have this size of the fragment, the I size per read. So one of the first things we're going to do is get some type of distribution of our scores. So do our reads look high quality mapping? We can extract that from our GA alignments object for either one of the reads. In this case, I'm going to do first and second separately. But we'll use mcols to get the metadata columns. So that's everything after these dashes, the 3 metadata columns. So I'm going to get the map Q. So I'll do mcols. And then I'll do $mapq to select the mapq column. I do that for each read separately, and I can just look, and we can see we have 40, 40. This is the top score we can get for subread. And then I can use our table function just to summarize how often do we see values in our list. So I'll give it the result from here, and then I can look at that here quickly, and I can see that 40 being a good score, most of our reads are 40, and then that drops off. Same for read 2. Read 1 and read 2 could have different qualities of alignment, right? And if we just do a quick plot using Ggplot now here, we can see that generally this doesn't look so bad. We have lots of high quality reads, high quality alignments here. What I would be concerned about is if I saw lots of zeros or 6s in this case. That will tell me there's something wrong with my data. I see mostly good quality alignment. This is our max, so I'm pretty happy. Now we have these reads already in paired end data format in R. We can then retrieve this insert sizes, and this is something which is super useful. Again, if this is probably the second thing people ask me to check in ATACseq. Since the properly paired reads will have the same insert size, so the distance from read 1 to read 2 is going to be the same as the distance from read 2 to read 1. We only need to look at one of the reads. Right? We know they're all properly paired, so I will just extract the reads from my first read from my GA alignments pairs object. That will give me this GA alignments object. And then for some reason, I've used element metadata here. It's the same as mcols. It's just another, this is probably deprecated. Mcols is probably the new one. But effectively mcols, and I will extract now the insert size column. So just to show you that, I'm basically trying to get this column here. This can be positive or negative depending on the orientation of the read, how far away. You know, that doesn't matter to us too. We just want the absolute fragment size, insert size. So we'll do abs, and that turns any of these values into positive. And if I just look at the head, I can see, you know, my fragment lengths here. So then I'll do the same thing again. I will use table on this to get the distribution of fragment sizes. And we're starting here, the lowest being 50. That's the minimum fragment size. We didn't set that, but that was the minimum fragment size in our subread. 51, 52, 53, 54. And it would go all the way up until 2000. Right? So we'll have a range of different fragment sizes. But because we know ATACseq should represent mononucleosome in part, we would hope that we will be able to see a peak around the length of mononucleosome covering fragments. So sorry to keep hopping back and forward. But this distance around a nucleosome, I believe, is like 137, 147 maybe. So we know how big, how much DNA can wrap around a nucleosome. Right? So by cutting either side of it, fragments which are basically that length, we'll assume came from nucleosomes. If they're packed too tightly together, we won't be able to get in between nucleosomes, but maybe we'll get around 2 nucleosomes. And then the distance of DNA, which is wrapped around them, will be twice. So we can also be able to find fragments which represent that. And that was actually the next thing which they did in their paper, and it's the next thing we can do to check quality. We just need to pass this now to Ggplot. So we'll do library Ggplot, data frame, and we'll give the insert, we'll create an insert size column of our fragment length sizes. I do names here as numeric because a name of a vector, even though these look like numerics, a name is always a string. You can't have integer or numeric names. So we have to convert that to as numeric, give the count, just to be sure I'm making that numeric too. And then I'll pass that to Ggplot, where I will have the X being the insert size. So longer insert sizes going across the X. The frequency of the different insert sizes on the Y. And we'll do geom_line. It's not super clear. It's about as clear as it was in the paper. We can see we have this big full spike here. And this is your open short fragments, open chromatin. And as we come down here, we can start to see this bump here. And if you look carefully, there's a bump there. Maybe there's a third bump there. Yeah, you really have to squint for that third bump. But it is there. And those would represent 1 nucleosome length, roughly around 147, 2 nucleosome lengths, 3 nucleosome lengths. Right. If you put that onto the log scale, it starts to become way more apparent. So if we just, same plot, plus scale Y continuous, and we'll do log 2 here. If you look at that now, you can see mono, di, tri, 4, 5. Right? So you can really see the evidence of this. And this is just looking at 1 chromosome. This looks, I think, identical to what the Greenleaf paper pulled out. So that's great. We can reassure this method we've reconstructed using Bioconductor is the same. But what they like to add onto it is really they wanted to define nucleosome-free regions. So they said anything under 100 base pairs couldn't have come from around a nucleosome. It does leave a large region where they're just not explaining. 180 to 247, they defined as being for sure mononucleosome length fragments. And then they did that also for dinucleosome. I think they actually probably labeled trinucleosome in that paper too. Okay. So we can make a plot and we can do something similar to what I think they did in their paper. Oh, there you go. So kind of messy, but 1, 2, 3, 4. We see 1, 2, 3. Remember, theirs looks a little cleaner because they're using every chromosome. We're just looking at one of the smallest chromosomes. If I'm not sure I advise this much anymore, ENCODE standards have updated since I first started writing this course. And ENCODE now will say, don't split up your data. Just keep it as one big open chromatin-like data set. But theoretically, you could now split it up by taking your paired GA alignment pairs object and just subsetting it so that only the ones with insert sizes less than 100, we can consider them nucleosome-free signal. Those between the ranges dictated from Greenleaf will say mononuc, and those between these regions will be dinuc. So I guess really anything which is, say this is the length of a nucleosome, anything which is 147 or above, we could assume is at least from one nucleosome, right? If I was to cut the open region here, I would have that bit of DNA wrapped around the nucleosome. If I unwind it, it's 147 long, right? So everything here could be anywhere between 0, I guess, and 147. Right? We would consider everything up to 147 to be nucleosome free. Right? We know it's not from a nucleosome. We might start to make the assumption once it's 147 and above, it comes from a nucleosome. So then the question is, why do they limit it all the way down to 100 rather than 100 to 147, just to be sure? Right? They don't want any of this overlap because they really wanted to be sure that we can consider it open chromatin. Was that the question? Is why isn't it 147 and below rather than just? So yeah, I mean, if it was much bigger than 147, we would start to at least know that we're contaminating our open chromatin, nucleosome free, sorry, with fragments which could have come from nucleosome. The issue is that there could easily be fragments in here which are over 147. You know, we're just making that assumption that they're probably belonging to. You know, it's not an exact science here. It's not exact, but it does a pretty good approximation. What a lot of people do, you know, there will be ones which have come from nucleosome free which end up in other ones just because the size of the fragment happened to be too long. But you should be able to clean up your nuke free by getting rid of longer fragments, right? They can't have come from nucleosome surrounding a nucleosome at that point. So you're saying, so whereabouts would that be here? So like here, from here to here. Yeah, if that was all open, hopefully though, we would have had a chop here, and then this region for sure we would have got some chops in, right? Transposase would have got in. So I guess that's the idea. But there still could be, for whatever reason, you managed to collect a fragment which didn't chop anywhere, and you managed to sequence that. The idea is that by getting the transposase concentration right, you know, you're going to break up your DNA in a reasonably efficient manner. If you really lowered that, you would get no breaking up, and then you would end up with really long fragments. Quite common is that people over transposase their data. When I first did this course in this class, there were a lot of people going, oh, shit, my data doesn't look like that. And it's because in reality, if you over too high a concentration of transposases, leave it too long, you get rid of all these other fragments. You end up really with just these short. You manage to break up all your DNA. Okay. So we can then separate out our reads or our fragments and then make a nucleosome-free mononuke dinuke. And then with that, we could use our track layer and just export the reads as a BAM file using export, give it the GA alignments pairs object, tell it the file name you want to write to, and just give it format equals BAM. Okay? And that will produce an indexed BAM file. I think one of the last things we want to do to actually output something is make a bigwig. Okay? So ATACseq is a little different from RNA-seq in that we were worried about splice junctions and stuff for RNA-seq. For ATACseq, we're not. It should be contiguously aligned to the DNA. Okay. So we can get our GA alignments pairs. We already have them here. If we look at one row, we can see we have 2 ranges. A little trick I can do with GA alignment pairs is if I apply them, or if I apply the granges function to that pair, you can see what it does is actually collapse it back down to a fragment. So if this is read 1, and this is the read 2, when I say granges, it actually goes from the beginning of read 1 to the end of read 2. You can see that here. Right? So that's actually kind of useful, assuming they must be from the same fragment. Now I can reconstruct the fragments as a granges, and with that, I can start to do some further downstream analysis. So the first thing I'm going to do is try and get an idea of my redundancy. To do that, I'm going to take my fragments from here, and I'm just going to use the duplicated function to tell me how many of these were duplicated, and I will use sum to actually sum up the number. So duplicated will give me true, false, true if it's duplicated, false if it's not. Sum, it will add up all the trues as ones, and it will give me the total duplicated fragments. I can get the length of the granges will give me the total fragments. And then to get a duplication rate, I can do duplicated divided by total. And so that will give me a duplication rate. There's a word which ENCODE uses called a non-redundant fraction. And that is effectively 1 minus this duplication rate. So this number here, 0.74, whatever, that is your NRF. That's an ENCODE metric. And that's a good score. There's quickly another QC we can check. We hope that not much of our data is too duplicated. Right? Given the size of the genome and the amount of open chromatin there should be, we wouldn't expect too much duplication. We can then quickly write this out to a bigwig. We've already got the fragments, right? So we created fragments here, so we can actually make coverage from the fragments rather than the original reads. And then we'll just do coverage on that granges. I'm going to weight it, so normalize it to the total number of fragments we had. I'm going to put in a million there, just so the numbers aren't too small. And then I'll export my newly created RLE list to this bigwig file, and then we can visualize that. Okay, so just as simple as going from 2 reads to 1 long read, which represents the fragment, and then creating coverage weighted by the number of fragments we have. So I think the final thing we're going to cover today, just enough time, is 2 more metrics. So ATACseq QC, it's written by a friend of ours, Zhu, we like her a lot. It's not the most memory efficient tool in the world. And that's just because ATACseq is hard to deal with. You have to hold a lot of memory, a lot of data in memory to do this. So I'm going to show you a little bit about ATACseq QC. sure she's very responsive if you write to her directly. But I warn you, it will take up a lot of memory. And you may never get to finish it on your laptop. It's just doing a lot. Two really useful metrics from this are PBC1 and PBC2. So this is your PCR bottleneck coefficient 1 and 2. And these are, again, originate from ATACseq, but have become, sorry, originate from Chipseq, but have become essential for ATACseq. Before I jump into this, another thing you can do is I showed you how to sample BAM files. So actually, you can often sample a BAM file down to 10 million reads and run this tool, ATACseq QC. It will work a lot easier. If you've got 100 million reads and you want to run this on your laptop, it's going to struggle. So they have this kind of workhorse function, BAM QC. So you load the library, a library ATACQC, and you can just provide the BAM file you want to process for ATACQC to the function BAM QC, and it will give you an object containing some useful QC metrics. So like here, I've just filtered down to chromosome 17 to save us some time. Effectively, this gives us back a list, and we can see the names of the elements or slots within our list. Some of them we've already kind of looked at. Mitochondrial rate, we can calculate that ourselves very easily. Duplication rate, proper paired rate, right? And that relates to having how many fragments are within your insert size. Non-redundant fraction, we know is duplication rate, 1 minus duplication rate. MapQ, we already gathered ourselves. IDX stats, we gathered ourselves. So all these metrics we can do ourselves, probably a little quicker than in the ATACseq QC. These two, though, we want to be able to get. So to extract them from our ATACQC object, because it's a list, it's going to be super easy. I can just do double square brackets and put this in the quote, or I can do $ and then that slot, and it will give me this metric. Okay. So just to talk you through what PCR bottlenecks about, really is it's aimed to identify PCR bias, overamplification, which can happen with some of these samples. And it's calculated as the number of positions in the genome with exactly one read mapping uniquely compared to the number of positions with at least one read. So if we have 20 reads, 16 map uniquely to locations, 4 do not map uniquely. Instead there are 2 locations, so they map to 2 locations, both of which have 2 reads will lead us to a calculation of 16 over 18. A PBC of 0.889. What we really want to know is what is a good score here. And ENCODE, this is the great thing about these big consortiums, because they run hundreds of data sets, they can kind of set top and bottom boundaries. And they would say 0.7 would indicate severe bottlenecking, 0.7 to 0.9, moderate, 0.9 and above, no bottleneck. Good. No, you'll just say it's acceptable at that level. So there's not much cleaning up you can do. I mean, people have tried methods for like removing duplicate reads. The issue with duplicate reads, and me and my old boss wrote a paper on this, which is still contentious now, but I think generally accepted, is that once you get enough sequencing depth, you start to get duplicates which are real. So removing duplicates can be a really dangerous thing. And if you can imagine you have a 36 base pair read on each strand, it means the maximum height you could have before you're a duplicate is 72. And as that limiting the maximum range of your signal became like a real problem for differential ATACseq and differential Chipseq. You just have a max, and then another one's still the max, but it's actually way higher since we removed duplicates. So there's not much you can do to clean it up, but if you had a sample which fell below this, I would start to think about maybe rerunning that sample. Most of the time, you're going to be, I mean, this is ENCODE data. You can see it's very close. most of the time with your data nowadays, you'll be quite high, a lot higher. And without slagging ENCODE, ENCODE got there first. So they were doing these methods when they were still developing. So a lot of their quality metrics are quite low. Pcr bottleneck 2 is a secondary measure, and it's calculated very similarly. So rather than, so it's the number of reads which map exactly one position uniquely compared to the reads which map exactly two positions uniquely. So not two or more, two positions. So then we could worry here that values less than 1 would be really severe bottlenecking. 1 to 3, moderate, 3 and above, no bottlenecking. As we can see here, we extract it the same way that we extract all the others. $, the element we're retrieving. And then we can see we beat our score there as well. Right. So this is really, it's a way PCR bottleneck of looking at the complexity of your data, library complexity. That's the end of this session. I'm going to have some exercises here, and they do start quite a low level, but you do end up creating an actual bigwig of the different types of signal. What is kind of cool here, as you can see, I mean, we'll come to it, but I'm not sure how much you know about it. Because you can kind of see the nucleosomes piling up either side of the TSS, which is often the case. You'll have like a -1 and +1 nucleosome. Also, I think in this one, we're also going to use some of Christina Leslie's data, which is kind of interesting because Christina Leslie's data is so much cleaner than the ENCODE data, but it is years on. What you can also see, though, is that Christina, as you get to it, she didn't have any fragment lengths longer than a certain range. So she must have cleaned up her data at some point to get rid of these ones we saw in the Greenleaf paper where they had, 1, 2, 3, 4, 5, 6, 7, 8, 9. You don't get that in modern day ATACseq. It's only the first 3. And I guess they do some size selection and clean it up quite a lot. You can actually see that for real in her data. Okay, so I'll leave you guys to this. I got to put all the videos up, so I'm going to try and do that right now. But I'll speak to everyone next week. No, Friday this week. Monday today. Sorry, cancel. If you have any questions, just let me know in between. I'm always here, available.